Okay. Hi, everyone.
Hello.
Okay, so today I'll present a talk, machine learning for the model-driven analysis of the brain. I presented that talk. So I prepared that talk originally and presented it at a conference that was organized by the for Mind and Institute for Mind and Brain here at USC. That was investigating the junction between neuroscience, so study of the brain, and AI. So I'm a faculty member of the AI Institute here. My focus of interest is in understanding how the brain works. So that's how these two topics comes together. The brain is an immensely complex system. You can get data on the brain that are very diverse. The brain can be studied at different scale, either spatially or temporally. So to be able to make sense of that, you need advanced analytic approaches, and that's where machine learning, AI, computer science, advanced computer science techniques are useful. So that's what I will be discussing. Feel free to interrupt me at any time during the talk. I'm happy to have it more, like I said, in exchange. And if there's too many questions, I don't go fast enough toward the slide. I just compress some part of the talk. I prefer to have it kind of so that you can relate to it rather than like no question and at the end you're like, what was that? All right. So if we're good with that, let's see. Okay. So what's my general objective? So I should mention, well, I'll read first the objective. So to develop a generic framework to address the problem of latent parameter inference in biological system. As I said, I'm interested about the brain, but you could frame it more generally as biological system because the approach is more general. And I put biological in parentheses because really you can apply it to any system that you can model. The idea here is to, if you can model, if you kind of have a generative model that produce the kind of observation that you would do on the real system, then you can use that kind of approach. So if you're not interested about the brain, but you're interested in some other system, you can still get the general idea of the talk and apply it. Latent is also underscored there. It's not too complicated if you can measure something directly on the system, right? The problem is when you cannot measure something directly. In the brain specifically, there's many reasons why you cannot measure everything. They can be ethical, because you cannot just like probe the brain of someone, right? And kind of injure the person. They can be technical. Maybe we have no means to get that value in vivo. They can be economical. Maybe it's just too expensive. And regardless of your system, you can think of these kind of limitations. It's not all variable that you can measure directly. Those that you cannot measure, you have to infer their behavior. So that's what is meant by latent there. Non-observable, you have to infer them. So through that approach, what we'll do is that we'll create a model of the system, and then we'll use this model to direct our analysis of the data that the system produces. When you have a model of a system, normally you have parameters. Through parameters, you can set how the model behaves. So if I take, for example, the heart as a system, my heart is not the same as your heart, is the same as your heart. If we have a model that represents how a human heart behaves, we need to have different parameters that can be fine-tuned to adjust it to how your heart behaves, how my heart behaves, how your heart behaves, right? So have a chair, there's extra chairs. So that's what we'll use. We'll use a generative model of that type. I'll come back to it in a moment. But in general, it's very simple, right? If I make it as a block diagram, it's like one block, an input, an output. So what we have in input, we have experimental data. So there's some experimental data that we can observe. So for example, in my case, I work a lot with EEG, electroencephalography. So that's when you put a bunch of electrodes on the head, and these record electrical signal. I'll explain a bit more what EEG is a bit later. But so you have some experimental data. They can be like if you're working with machines and robots or whatever, they could be like a vibration sensor, right? Whatever you have.
One question. So the data part, I got it, but the inference is happening in probably some kind of a user interface, and there could be a model that we will be building on using that model.
It's not a user interface. I'll come back to it. So if you have a model and you want to understand how this model works, so a model that operates in time and that have produced some kind of observable variables that you can measure in time, so you have signals, right? Was there a question? Okay. So normally how you would model these kind of systems is through a set of differential equations. These differential equations will have different parameters that you can fine-tune so that the model operates in a space in a part of its space where it produces the specific behavior that you expect for that individual or that machine. Does that clarify it? Yeah. Okay. So the experimental data is what we have. What we want is estimating some useful, I say parameter, but some useful value. In my case, maybe like in the brain, there's some specific process that I cannot measure directly through EEG. I could measure it if I was to like implant an electrode in the brain, but I cannot do that because that would be damageable, and right, you don't want to do that. So maybe I can try to infer that. So that's what I want. I don't know, like how the brain behaves in that specific region. What I need is that block that do this inference, right? So that's very large level. I'll present two example of use case. I'll keep them across the talk just so that you can kind of relate to something practical. But you can flip them with, like, if you're doing some research and you have a problem, you can kind of frame it in context of your problem. So in that first example, it will be an example using EEG. What we have is non-invasive brain recording. So as I mentioned, you put some electrodes on the scalp of someone. These electrodes are at the surface of the scalp. What happens is that there's a population of neurons called pyramidal neurons, which tend to have their what we call their apical dendrite, all aligned perpendicular to the cortex. The cortex is the surface of your brain here. So because they are all aligned, they tend to, they create small dipole. Each cell creates a small dipole between the tip of its apical dendrite and here, which is what we call the soma of the cell. It's kind of like where there's all the machinery of the cell. So it tends to create small dipoles there. And since all these apical dendrites are aligned, all of these small dipoles sum up together and create a relatively large dipole that creates a field that is large enough that you can actually pick up at the surface of the skull. That's basically what you get with what you capture with EEG. When you have a bunch of these neurons that are all receive information at the same time, they all create these dipoles, create a large equivalent dipole, and the electromagnetic field kind of propagate all the way to your electrode and you can capture it. It looks a bit like that. The signals that you have completely at the right of the slide. So it's kind of like noisy signal that goes up and down and up and down, and we can interpret these. Yeah.
So since you mentioned that there's an electric field being created, so it's not just one region where the electric field is being created, it's throughout
the brain.
So there's always a chance of one electric field being interfering with the other. And is there any way to differentiate
mix that? So that's kind of like a mixing problem. So if some of you work with something like ICA, where you try to unmix different source of activity, you have this problem there, right? Because you have a huge number of, it's not even a huge number of of brain regions. It's basically a continuous map of activity, right? And it all propagates, and then you have a mixture here, and you have to unmix it to kind of localize the activity in the brain. So that's the problem of source reconstruction in EEG. I don't think I talk about it in that presentation because it's not necessary for the topic, but there's definitely a field of research that's developed approach to do exactly that. Yeah, it's non-trivial. It's not because you get some activity here that it comes directly from underneath, because there's all that mixture of activity. Okay, so that was what we have, right? That's the EEG. What we want. So for my specific use case here, I want to evaluate something that is called the excitatory-inhibitory ratio. I'll just call it EI ratio from now on. So in your brain, you have you have three types of cells. One of these type of cell is the electrically active responsive cell that we call the neurons. There's two big groups of neurons, the excitatory neuron and the inhibitory neuron. So the excitatory neuron, when they talk to another neuron, they tend to excite them to make them more likely to also talk to other neurons. The inhibitory neuron, they tend to inhibit the neurons that they target. So if you have a neuron that was to emit what you call an action potential or a spike, that's the way that neuron communicate messages. So it was about to communicate to generate a spike, and you have the other neuron that say, hey, so that's your inhibitory neuron that kind of clamp the activity of the other neuron. So these are inhibitory neurons. So in the brain, you have these two populations. The balance between the two, or the ratio, is very important. If you have, for example, two excitation, you can have runoff activity, which would cause epilepsy, for example. And it's been observed that this excitatory-inhibitory ratio is off in many neuropsychiatric conditions, for example, epilepsy, schizophrenia, autism spectrum disorder, or ASD. I'll use the acronym ASD for the rest of the talk. Attention deficit hyperactivity disorder, or ADHD. So for that use case, I have EEG from people that have autism, some people that don't have autism. And I want to find a way to evaluate their EI ratio from EEG. I cannot measure it directly, that ratio, because that's associated to specific neurons. I cannot go in there and record specific neurons, right? So that's what we want.
Quick question: Are you doing anything with fMRI?
Not in that talk. We do have projects that use fMRI in different so I'm working on multiple projects. So with some collaborator, we are looking at fMRI. If you're interested in talking about fMRI, we can definitely talk a bit after the presentation. Okay, so that's my first use case. The other use case, so I'll use something a bit different. It's still, I'm interested about understanding the brain. It will still be related to the brain, but it's more, here we'll go more peripheral. So we'll model the heart. Well, actually, we'll model what we call the autonomic nervous system, which controls the heart. So what we have, it's non-invasive recording of cardiac activity from electrocardiogram. So that's basically the same thing that you had from the EEG. So you had the electrode on the head. So instead, you place them there. And then instead of recording brain electrical activity, you record heart electric activity, right? So ECG, you've seen them, unless you've never been exposed to television, you've seen ECG. Like if there's some show where someone arrived at the hospital, they plug them on the machine and beep, beep, beep. So that's your EEG machine, right? And it's probably look a bit, so this one is a cartoonish version of it, but it probably look like, unless you have the heart is defibrillating or something like that, but like a typical ECG would look a bit like that. So that measure cardiac activity, so it's a perfectly fine topic of research, so you could be interested in cardiology and just study how the heart works. In my case, I'm more interested about the brain, and so I'm interested how the neural system So I'm interested in a division of the nervous system. So you have the nervous system here at the top. It's divided in two big branches. Your central nervous system, so that would include your brain and your spine, and your peripheral nervous system, all your nerves that control your movements, right? And also that sense. So if I touch something, I'm able to sense it. There's a nervous system that connect to my brain, right? That peripheral nervous system is divided in two branches. The somatic nervous system, so that's the system that allows me to, it's a volitional part of my peripheral nervous system. So if I say I want to get water, I'll aim for the bottle, I can control that. The other branch is the autonomic nervous system. That's the branch that is not, that we don't control voluntarily. So I'm not constantly thinking like, okay, I have to beat my heart, I have to beat my heart, and I put attention to beat my heart, and if I want, I can stop and restart, right? I can do that with my breathing. Breathing is a bit between the two. It can go like automatically, or it can be controlled, but the heart is completely autonomous, right? So that's this branch, the autonomic nervous system, control the heart. It has two subdivisions, the sympathetic and the parasympathetic. The sympathetic is the one that is activated when it's the fight or flight system. So if something is like arousing you, like you have to react, you have to defend yourself, you have to run, you have to, I don't know, you have a stress response, that will increase your sympathetic activity. When you are relaxed, you can kind of just rest. It's the parasympathetic system that will activate. And the system, the autonomic nervous system, balance the two. And in ASD, this balance, this control between the two is dysregulated. And because this impacts the cardiac activity, you can record ECG and use ECG to evaluate dysregulation in the autonomic nervous system. So that's what I'll be interested in the second use case. So I will use ECG to assess autonomic regulation. So that's what I have, ECG. What I want is to determine ASD diagnosis. So this is for infants before they are at an age where we can actually diagnose them with classical tools, behavioral tools, basically. So how it works is I have ECG activity. From ECG activity, I can derive a measure of autonomic dysregulation. And from that, I'll use that to diagnose ASD in infants. That's the use case. Okay. So we have two specific examples that will be used throughout the presentation, but the framework is general. What you have some signals. So here they are both biosignals because that's what I'm interested in. But again, it could be like sensors on machine or whatever. As long as you can develop a model of how your machine produce these observations. And I have some variable that I want to assess that I cannot just measure directly in any simple way. So if I generalize a bit that problem, so I'll take my input, put them in like a matrix form, refer to it as a matrix X, like you have like in machine learning, right? You put all your features in there and you have this matrix X. And what I want is basically parameters or variables. I'll represent them as theta. And now I have an inverse model. So I have a functional relationship that links my X to my theta. I use for my functional relationship -1 exponent, right? To highlight the fact that this is an inverse model. Why I call it an inverse model is because it runs against the flow of causality. So a direct model tends to run in the direction of causality. So if I have some mechanistic understanding of how a process generates data, I can, if I change the parameter in these mechanisms, I'll impact down the line the resulting observation, right? So if I change theta, and that will cause a change in observation. So that would be the causal direction. That would be my forward model. Here I have an inverse model because I start from the output, so the causal result, and I try to infer the cause, if you want. Okay, so so you could try to address that with a supervised learning approach. So in that case, what you have is a large quantity of recording with both the value of x and theta. So it's supervised learning, right? So you need to know the output associated with a bunch of your input so that you can train a system to learn that relationship. So if you want a way to estimate the parameter theta here, so for example, my EI ratio, giving matrix X, for example, my EEG, I can use machine learning. So in that case, a supervised learning approach to train a neural network to estimate the function, my inverse function there. Why I can do that? Well, because deep neural networks are known to be, you have a paper here that is one of the papers that demonstrated that they are universal function approximator. If you throw enough data at them, they'll learn the mapping. And then you can use the mapping with new data, right? So that works fine, and that's a relatively simple, like nothing is simple in life, but it's comparatively simpler than other approaches because there has been a lot of research, and like you use SciPy, you can easily implement a supervised learning model to test a model, to develop a model like that. There's a limitation, however. One of them is that it requires a large data set of both the x and the theta, right? If you don't have that, it's a no-go. You also have to have a way to measure theta. So that's fine if it's measurable, but if it's not measurable, what do you do, right? You don't necessarily have that. So an illustrative example of that, taken from one of our previous study at the lab. We have ECG recording from infants at 3 to 6 months. We compute some features out of that. So these are classical features for ECG. I put the list here, but they are not, unless you're interested in ECG and or cardiac activity, they are probably not super important for you. Then when you extracted the feature, you have a classifier. Again, there's a bunch of classical classifier. In that case, the one that performed the best because it was a relatively small sample size is the key nearest neighbor. And then you do classical assessment, sensitivity, precision, accuracy. So I'm sure you're familiar with that kind of system. There's nothing super fancy there. It's just to find the right features, and like it's always in the implementation, right? You can spend like a year like making sure that everything is good, but like technically it's not complicated. It's in terms of like the science of it, right? So that's one approach. It has some pros. As I said, it's kind of relatively easy to deploy. Again, you can spend like a year cleaning your data set. You try to compute the features, the code to compute the feature bug. You have to debug that. You test multiple classifier. Maybe you don't obtain the result you want. You realize that you have like, so there's a lot of analysis that goes, and you can spend a lot of time implementing that. Yeah.
Now, is your research designed to like partner with the medical school here on a specific use case, or are you trying to develop generic tools for medicine in general?
A bit of both. I have active collaboration with the Department of Psychology. I'm affiliated with the Carolina Autism Neurodevelopment Research Center. Psychology has a big representation there. Also, School of Medicine. So yeah, it's always linked to actual use case. Being more from the engineering side and the computer science, of course, I'm developing more the approach. So yeah, a bit of both. Yeah.
You did in the previous slide that you talked about autism, right? Identifying autism in the early stages. So those features is being you where is the reference point in terms of autism?
I don't know if that's what you're getting at. I'll say that, and if I didn't, then if it doesn't answer your question, ask again. Note that I said you need to be able to measure both your X and your theta, right? And I said, well, so in that case, I cannot measure my theta in ECG at 3 to 6 months. We know how to diagnose autism at 2 to 3 years of age normally. So it's not available here. I have to have a method to estimate it. But I can actually build a data set where I have access to that. I just have to wait a year and a half to a year and then measure it. And then, so these are prospective longitudinal data set where you collect the ECG at 3-6 months, and then you follow these kids until they have a diagnosis at 2-3 years of age. And then you can say, okay, now knowing what diagnostic they will have, can I train a machine that would be able to predict that in a new kid? Does that answer your question?
Is it basically that we have a recording of a normal human brain? That would be like your reference point, and then anything like if they are thinking that that's going to be the sample, then we'll compare it with it, and then if this is not matching with a normal human brain, then there is a chance of autism.
Yeah. So you can do that as a classification problem. Two group has autism, doesn't have autism. Or you can do a regression problem where you have a scale of autistic symptom severity. Even in neurotypical population, we have a different level of autistic traits. And then when you have too many, you pass some threshold, and then it's okay, you have an autism diagnosis because it starts to kind of make it harder for you to integrate in society. But so you can have also kind of a measure of severity that goes from almost no autistic trait to a good amount, but still neurotypical, to a certain amount with the diagnosis, or like high severity autism, right? So easy, quote unquote, to deploy in the sense that the approach is known. It's just like it may require a lot of work, but we know how to do it, right? You can read books. It's also an easy approach to motivate because it's tightly linked with biomarkers, which are easy, right? Everybody understand like, oh, you have a severe condition. We want to be able to better diagnose it. We want to be able to better monitor, to offer better care. It's easy to sell, right? So these are pros. In the cons column, requires a large data set for training. There's techniques that can allow reduce the amount of data, so transfer learning, foundation model, right? Training to recognize, to process ECG or EEG data on large amount of these recording in other problems, making sure that these classifier understand how to extract meaningful feature out of these data, and then fine-tuning it in smaller data sets. So you could always do that. But in general, requires more data. The more complex the classifier, the more data it requires, right? And we don't always have that. So in biomedical, so you have, for example, rare disease. So autism is a spectrum. There's people that have this diagnosis for different reasons. Some are rare genetic mutation. You may have 100 person in the world that we know that have this genetic condition. So forget having like a million EEG recording for a rare condition where you have 100 people in the world that have it, right? So biomedical application have intrinsically, in some case, low sample data sets. You just have to deal with it.
Professor,
yeah.
One more question on this one. But when you say the significance of biomarkers is easy to motivate, So, like say we have a recording and then you're extracting the features of it and then you're detecting it. But how does the biomarker come into this?
So I record your brain. So let's say we had no ways to diagnose diabetes, but we know that there's such a thing as, it's not the case, right? It's easy for us to do, but let's imagine thought experiment. We know there's such a thing as diabetes that can have important consequences if not regulated. We have a strong motivation to diagnose it, but we don't have a clear way to do it. And then I come and I have this hypothesis that maybe from diabetes modify how your brain waves are structured. So I'll say, okay, I'll record your ECG and I'll analyze them and I'll develop a metric, a measure that is correlated with your probability of having diabetes. So that's my biomarker. It's a variable that correlate or predict the presence of a condition that we are interested in diagnosing, right? So if there's a disease that causes a lot of pain in the population, costs a lot of money in the society, in the health system, and I say, well, I have this project that will allow us to diagnose it earlier, better monitor the people that have this condition to provide better care and reduce a lot the health expenses. It's easy for me to go to NIH and make a case for it. You should give me $2 million to do that because that's a $1 billion problem. You know, in that sense, it's easy to motivate. So other limitations that if you want to use that, you have to have a way to measure the desired parameter, so the theta, and it's not always available. In that case, it's a temporal thing, right? I may be not able to evaluate it now, but I can actually build that data set because I can just wait for the kids to age, do a proper diagnosis, and then I have retrospectively that data set that allow me to do that. But in context of what I was saying about the brain, if there's something that I just don't have the technical means, or ethically it's not possible to measure that in a live human, and when the human is dead, the process doesn't exist anymore. It's like the brain is not functioning anymore. I have no way to measure it. That's a no-go, right? It does. So the other thing is that it does not model the mechanism. So what it tells you is that, oh, I mapped a relationship between observation and some parameters, and I can predict them, but I have no idea how the parameter caused these observations. I can predict them, but like, what's the mechanism? How does it work, right? How does my excitatory inhibitory ratio impact the activity of the brain in a way that you have autistic behavior? So that doesn't help me at all for that. It's useful for biomarkers. So there's good accuracy to predict that mapping allows me to have clinical applications. So that's very useful. But if I want to go one notch further and really understand the process to develop, for example, drugs, it's not sufficient for me to know that, oh, that person had that condition. I have to understand how in the brain the behavior of the cell get dysregulated, so that I know what molecule to target through medication, or how to modify the system. That understanding, I don't necessarily get it with that type of approach, because it's not mechanistic. It's just statistical in nature. Often do not translate into clinical use, so there's reproducibility issues. So the good example of that is like 10 years ago, there's a group that published studies where they were, among their analysis, they were showing that they had recording of infants at 3 months of age, and they had like a 100% accuracy in predicting autism at 3 months of age from the EEG. I look at recording of infants at 3 months of age, and believe me, it's not beautiful, and it's very hard to correlate them with autism. And it's not just because I don't know how to do my job. It's just like there's a lot of noise in these data. And how do you know that that it's not just me that don't manage to do that, and they are so much smarter than me that they can do that? That 10 years later, there's still nothing in the hospital. Like if that was a real thing, reproducible, like 10 years ago, they would have come on the market, put a product there, and now at 3 months of age, we would put a EEG net on the head of baby, and we could say, okay, this baby will have autism, give them some therapy or approach to help them compensate earlier, correct earlier that trajectory, right? It's not there. And there's no other study that reproduce that kind of results. I said, well, yeah, okay, your machine learning gave you good results, but like probably you didn't properly cross-validate. There was probably some information leakage between your training and your validation set. So you ended up having 100% accuracy, but it's not a real thing. So you often have that also in these studies, unfortunately. Okay, so what I propose here, it's another approach, which I would refer to as model-driven approach. In that case, what you have the X value. So in that context, for example, EEG recording. The other thing that you have that you didn't need it in the other approach is an understanding of the process by which theta, your parameter, generate, cause, influence the generation of your observation X. Because that's what you'll model, right? So if you have these two things and you don't have the theta, so theta is a latent variable. You cannot measure it directly. You just don't have the mean to measure it directly. And what you want is a way to estimate these theta given your observation. Then you can use that general approach. So note that in blue here, understanding the process by which theta generate or causes X, that's your forward model. How does your theta and input is functionally related to your output X, your observation, right? Now, what you want is a way to inverse that, so to do your inverse model. So starting from the data, passing them to the inverse of your forward model to obtain the theta. That's my inverse model. So in a nutshell, when you have a forward model, I can pass a theta there and then generate the observation, check, are the observations similar to my recording? No, change a bit the theta, resimulate. Well, am I closer? Yes, okay, I'm in the right direction. You basically can do a gradient descent, right? If your data are well behaved and all, it's an optimization problem. Travel your theta space and find the place, the combination of parameter where your X is the most similar to your actual observation, and you'll have an estimation of your latent parameter, provided that that your system is well behaved, that there's not it's not underdefined or overdefined. Yeah. Oops. All right. Model-driven analysis try to estimate latent parameters, so your theta, giving some experimental data X. So that's your problem of finding that inverse model that gives you, for any value of X, you get the theta. So you can expand that into that system here where you have a forward model. From the forward model, you simulate some observation, you compare them to your actual observation, you can compute some loss, and your goal is to minimize the loss, so you basically have an optimization problem there, right? So parameter estimation, the inference is an inverse modeling problem, so it's anti-causal, so that's my inverse problem. I can use my forward model to develop a solution for my inverse modeling problem. Note that also your forward model, so that functional relationship, normally it's not just a blind mapping. It's not something that you train, for example, using machine learning. Typically, it's a biomechanical model. So you know how the brain works, or how whatever the system you're analyzing works. You study the processes. You have laws of mechanics, laws of electromagnetic, laws of chemistry, or whatever the field that your system can be described with. And you use these laws to define how, giving some parameter, you observe some observation. So that's where you inject your knowledge, right? So that's you inject all the knowledge of your field into your solution there. Yeah, so inverse modeling can be.
But if your data was like blood chemistry, you could get a snapshot of it, right? Like you could say my blood protein is high, my heart rate's probably higher than normal.
Yes, but so that's one way that you can observe the system, right? But it depends what you try to predict. If I'm working on the heart, and what I try to predict is there some default in the valve, which is known as a specific condition in cardiology. Maybe the proteins can inform that, but maybe not, right? Or maybe it's one of the parameter and you have to consider a bunch of parameter. Maybe you would need to observe that in continuous, like every 15 minutes through 1 month, to have like a picture from which you could extract, and it's not convenient to every 15 minutes do a blood test, right? There can be all sort of reason why you cannot measure it directly. If you can measure it directly, forget about that. Just measure it.
Right. No, I'm just thinking you could actually work in both directions. In some cases.
Yeah. I mean, the more observation you have, the more you'll be able to constrain your model to get to your answer for sure. But so that's the kind of expansion that I'll describe a bit more. So there's three important elements here: the loss function, forward model, optimization algorithm. So we'll start with the loss. So hopefully you're all familiar with loss function. It's used nowadays everywhere in AI machine learning. So there's multiple loss functions that are used in the literature that are better adapted to different type of data, different type of problem. For biosignal, and in general, the mean square error is probably one of the most common, right? You have some observation, you have some estimated value, you compute the error between the two, take the square, sum all of these error, and take the square root of that, or no square root, don't even need the square root. But so the problem that we have here in biosignal is that computing a loss like a fitting error between signal is sometimes a bit problematic. And I'll give an example. So that's, again, it's with ECG. So here I have a profile of how my loss varies as a function of some parameter here, heart rate. That kind of loss profile, I'd say, I'd characterize it as a crack in the floor, in the sense that there's no gradient, right? It's basically a flat surface. And just when you reach really like very close to the optimal solution, it does like a crack, right? So let's say you want to optimize that, and you try to do a gradient descent on that. Very hard because you don't have gradient that helps you anywhere except in the close vicinity. So you would basically have to go randomly until you find by chance that you get by chance very close to your optimal solution, and then your gradient descent would help you. Now you could say, well, it's not too bad. I can like evaluate at 50, 51, between 50 and 90 at every values, and I'll get close enough and do gradient descent. That's true in one dimension. But if you are in two dimension, that becomes a point in a square, right? And as you go in larger dimension, that becomes impossible to find if you have that kind of profile, right? So why do I have that? That's a real loss curve, right? Well, why do I have this in that case? I have this in that case because my signal has high amplitude, narrow duration events. So my ECG peaks here, they are very high amplitude and they are regular. So as soon as I don't have just the right frequency in my model, as time passes and soon enough, the peaks get offset, and now the loss is completely. It doesn't matter that I'm close. I'm as soon as these peaks are not aligned, the loss goes super high, and then it doesn't make any difference, right? So in a context like that, you cannot use directly the loss. So there's different approach to solve that. A classical one is just to use to compute features derived features out of your signal, not just to compare directly your signal, but engineered some specific feature that characterize the important characteristics. Sorry, I'm a bit reading my vocabulary here. Characteristic of your problem. So I expand it with two blocks. So I get my variable x. Now I compute some set of feature f. I do the same thing when I have my simulation. I compute f prime, which is the same set of feature. I do, instead of doing my loss between signal, I do just loss between different set of feature. And that quite often works better because the feature are engineered in a way that have a better, less of that profile where you have kind of like as I said, like a crack in the floor in your profile of your loss. Features, these depend a lot on your applications, right? So for ECG, there's a bunch. You can find them in state-of-the-art paper on your problem. So for ECG, there's a bunch of heart rate variability features that you can extract using well-known toolbox. Same thing with EEG. There's a bunch of different features that have been used for the last 50 years to do statistical analysis on EEG. So you can derive the ammeter using what we call evoc-related potentials. So how the brain systematically responds to a specific stimuli, or the power spectrum. What band of energy your brain responds the most in some context, or functional connectivity, how different brain regions talk to one another, right? You can extract these values and use that as features. Or maybe you have no idea, it's a new problem, there's no features, you don't want to develop features, then you can use deep learning if you want to kind of automatically try to learn meaningful encodings of information from your signal. Classical one, the autoencoder, where you pass your signal through a funnel shape with some multi-layer network, right? With the bottleneck in the middle, and then you do the opposite operation, and you train that system so that it is able to reproduce the input signal, right? So you pass your signal. You always have the label what you try to predict, because it's the same thing as you put it in input, right? So there's no problem there. You have both the input and the output. You just have to use the computation to train that model. So now when that model is able to reproduce faithfully the signal in output, it means that all your information got squeezed in your middle layer, in your bottleneck there. It means that the weight or the information in that middle layer is encoding all your information in a small set of weights, right? So you can then use that as a feature. Doesn't help you with okay, what that means, but it can help you to as a feature, right, for fitting stuff. You could also use embedding from pre-trained time series models, so transformer, domain-specific foundation models. So these are popular these days. Once you have your features, maybe not those from, well, you could try to do that on those from deep learning, but typically you would do that with meaningful features. You may want to do a sensitivity analysis, because if you want to predict from an observation a parameter, the only way you'll be able to do that is if the parameter influences the observation. Maybe changing the parameter has no effect whatsoever in what you're observing. If that's the case, there's no way you'll be able to predict it or to learn that association, right? So you can do a sensitivity analysis where you look at just in simulation how modifying the value of my parameter affect my simulated output. And if there's not much effect, forget it, it's unlearnable. So before trying to learn something, right, check that it's learnable, else you're losing your time. So that would be done with the sensitivity analysis. So we've seen the loss, feature computation, forward model, that's very domain specific. For EEG, we can use what we call neural mass models. I won't go in detail because I'm a bit short in time here, but it's basically a system of differential equations that simulate neural activity, average neural activity in brain regions. When you have, so how these work is basically you have the incoming activity. So you have one brain region, the other brain region that sends activity to that region, right? That activity is convolved with a kernel that describes how the synapse integrate information. So that gives you the average activity, if you want, in these population, and it's fed to the next population. I probably didn't give enough information for you to unpack that. Just I'm short in time, and it's very specific to neuroscience, so maybe not relevant for most of you. But in short, when you have these neural mass model, you can then put one neural mass model in each brain region, connect them with what we know of the connectivity between brain region, and then simulate a whole EEG, a whole like human EEG. So that's your forward model, and then you can use it in that framework. Last block, optimization. There's a bunch of optimization approach to optimize the system, deep neural network. So with one of my students, we use LSTM, transformers. One system called DeepBase use LSTM also, and GRU. Reinforcement learning can be used. I'll show an example in the next slide. Bayesian inference is used a lot for that kind of application. So you don't necessarily need to go the machine learning way. You can also do Bayesian inference. And evolutionary algorithm has also been shown to give some good results. Here I have just an example with reinforcement learning. So I have a model of my, okay, I have my ECG recording. So ECG recording generate these waves, stereotypical waves that we call PQRST complex, because it's each wave is named PQRST. So you can decompose your ECG in many PQRST complex. You can stack them to get an average PQRST. So that's my data that have been pre-processed. On the other hand, I have a model of how I can, a model of this PQRST complex. here using log normal, a sum of log normal functions. There's a whole theory on why log normal function, how to interpret these parameters. But I have basically my prototype of PQRST complex that is parameterized with a set of parameters. Then I can use deep reinforcement learning, for example, in that case, where I have a system that try to, so it observes the data, it observes the current fit of the model and the error, and it try an action. So an action would be to modify a bit, to go in one of or the other direction of that parameter space. Depending on that action, it will observe its environment, see what's the error. So the inverse of the error will be its reward. So the lesser the error, the better the reward. And based on that, it will update its policy of, given some observation, what should be the next best option, next best action, right? So using that approach, and you can train your system and show that you're able to do inference on these parameters. So that's the whole framework. We tested it in using so Bayesian deep learning, basically using. So what you do in Bayesian deep learning is that you have your forward model. Since you have your forward model, you can draw randomly from prior distribution your set of parameter and generate outputs, right? The observation, the EG. So I randomly draw a bunch of combination of parameter, generate a huge data set of these parameter give this EG, this parameter condition give this EG, right? And I can feed that to a machine learning model, which learns this association. And that's basically my inverse model that has been fit by machine learning. Then I pass new experimental data and say, okay, now what were the parameters that would have produced that EG? We did that with DEPA. We saw that some parameters are well learned, easy to learn, some nothing to do, they remain flat. That's work in progress, so maybe this curve will still improve in the future, but we have some very good correlation on some key parameters. Other parameters are, they are not learnable. It may be improvable by how you frame your problem, right? You can have parameters that are expressed in a way that your model is not well behaved. So you have multiple combination of parameter value that give the same observation. Sometimes just doing a transformation of your parameter space can solve that kind of problem. So that's still something that we have to investigate. So in conclusions, I'm about 1 hour in. I imagine that's about the duration that I'm supposed to take. So you can use generative biophysical modeling, depending. So bio, if you're working with bioscience, just physical or chemical, or depending on the type of system you're modeling, right? But you can use generative models and machine learning to combine your knowledge about the system, so in my case, the brain, to estimate latent variables, so things that you would not be able to estimate directly. More effort are required in different directions. So one is that field is kind of like different field developed things that fall, that you can kind of wrap into that kind of approach. But these different subfields, they don't talk to one another. Sometimes they don't even know that, oh, there's another field of science that do exactly the same thing that we're developing, right? So it's like it's been developed in very siloed approach, and that needs to be a bit integrated. We need to develop more robust approach for inference parameter in dynamical system. So doing parameter inference in dynamical system is very hard, because dynamical system can have chaotic behavior. So there's part of their parameter space that will have chaotic behavior, part that will be well behaved, And then it's like, oh, your system works, and then bam, you just increase a bit the parameter, and then like it's unpredictable, right? So that's very hard if you throw machine learning at that, because when you're in the chaotic region, there's basically nothing to learn. Make model-driven analysis easier to deploy. Right now, if I have like a graduate student that is interested in that approach, I cannot just say like, oh yeah, read the tutorial of model-driven analysis and try that and come to me with results. So it will be like a few years of training the student in the approach so that they kind of wrap their head around it. Develop didactic materials, kind of linked with the other, right? You have to have like good way to teach these things. So develop didactic material to allow students to get familiar with these approaches. I think that's what remains to be done in that field. That's it for me. Is there questions?
Are you using LLMs, or you have your own tools local?
So we didn't use LLM yet. We use LSTM. We use transformers. So I'm more and more interested in foundation models for time series. So foundation model for EEG, for ECG, as a way to encode efficiently these signals. Not done that yet.
Okay.
Other question? Yeah.
This might be just being out of the medical space, but
Sure.
The whole point of you trying to do this is to detect a lot of, let's say, brain disorders earlier on in their development. But even then, what would you do with that information? Because I've always been told you don't want to mess with the natural development of someone that young, if it's abnormal or normal, until they get to about 3 to 5 years of age, and then you can start doing something about it.
Yeah. So approach would be gentle, and like there's approach that we're pretty confident that cannot do wrong. So if you say, oh, there's this kid, they need an enriched environment in terms of stimuli and social interaction because they'll have more challenge in that direction. So we want to kind of overexpose them a little bit to interaction. There's probably no way to go wrong doing that. If you're saying, oh, I'll use that experimental drug to change their receptor, that's probably a big no-no. And how these things are developed, you go from the less vulnerable, less sensitive population, and then first in animals, like low-level animals like rodents, then like maybe monkeys, then like there's different level of clinical trials before you arrive in using a drug in infants. It's like you're like 20 years into the testing, right? But yeah, but why you would want to do that is because if you take something like autism, it's a neurodevelopmental disorder, right? Meaning that it unfolds as these infants get older. So you can think of two trajectories where when they're born, they're probably already a bit different, probably start in trilateral, but they are much closer. And as they'll develop, they'll kind of diverge. If you can catch them earlier where the divergence is smaller and kind of just nudge them in the right direction, it's probably easier than when everything has crystallized, right? They are also the brain of infants, never children, is very malleable, very flexible. By the time we're adult, we're kind of rather crystallized. So the earlier you get them, the more chance you have to kind of gently nudge them in the right direction, mostly through behavioral kind of therapy. Yeah, that's good. Other questions? A lot of people like on the edge, ready for the door.
I have a lot of interest in fMRI.
Let's discuss after.
I read all the Cal Berkeley studies on that a few years back, and when I worked for Verizon Federal, and they were working on mobile extensions to fMRI. So being able to wear a headset all the time. So for
fMRI,
like for soldiers and military applications as well. So
fMRI. I'm
Yes, sir.
surprised by that.
yeah,
So there's FNIRS, which would get you something similar than fMRI. So FNIRS is a functional near infrared spectroscopy, where you shine basically infrared light through the head, and like through the diffraction of the light, you get the same type of signal that you get with fMRI. So it's like the blood oxygenation. But fMRI, I was not aware of it, and because it creates a huge electromagnetic field, right? So if someone wears something and pass near metal like this, right? So even when we do,
I think
yeah.
it's doing measurements in real time and then decoding those later.
oh, okay,
So
yeah, okay.
It's you know, I guess latent analysis collection. So,
Yeah, you can probably do online analysis. That's
Yeah.
can be interesting.
all right.
All right, thank you, everyone.